Yes, we are. Okay. Assalamu alaikum wa rahmatullahi wa barakatuh. We are back after a long journey, and inshallah, we're going to start, another journey. This time it's about biochemistry. So inshallah today I want to learn, I just came back from the class about the biochemistry class, so I want to learn more about the, what do you call it, carbohydrates metabolism. Carbohydrates metabolism, yes. So I'll just try to open some books, inshallah, and see how it will go. So, where is it? I think it's in education. Oh shit. Let me just reconnect the USB. Hopefully it will work this time. Education, there we go. Okay. Medicine. Engineered metabolism. I think there's an issue with my camera. Hold on, let me fix it. Okay. There we go. Okay. So let's study about glycolysis, breakdown of sugar, which produces from glucose we'll get down to pyruvate, and let's see how does that happen. Bismillah.
A little definition of glycolysis is they're going to oxidize a molecule called glucose. So glucose is a 6-carbon molecule. It's basically a monosaccharide, which is just some fancy word for a sugar. So glucose is a 6-carbon molecule, and we're getting that from our diet. We'll talk about how exactly we're getting it from our diet and the digestive system. But for right now, we're just going to say that we're bringing glucose into our cells, which is again is a 6-carbon molecule, and we're going to oxidize them through various steps, about 10 steps, to eventually convert that into pyruvate, at least 2 pyruvates, which is 2 3-carbon molecules. But the question is, how can we get this glucose, this 6-carbon molecule, into this cell? How do we do that? Because glucose, so again, what is this molecule right here? This molecule right here is specifically called glucose. Let's actually write it in red, make it nice and curved. And this is called glucose. So glucose is our 6-carbon molecule.
Obsidian, my favorite note-taking software. I just had an upgrade.
Molecule, we're denoting that by these circles. 1, 2, 3, 4, 5, 6.
Let me just pause this for a bit and let me open up my notes in the software. Okay. So, yeah, basically, I'm gonna create a new one. Create a folder. No, not folder. Folder. It's not this biochemistry called carbohydrates. Please. Okay, so let me just review the material that I need to study. Okay, I have like this living cut here, living cut, and cut my cursor like this. Okay, I'm just gonna, this is a very, you could say an in-depth. It's not an in-depth, it's just review. But I'll just start with someone, teaching me the material. Then I'm going to look into the books and references and whatnot. That's my study method.
The thing is, though, glucose is actually a water-soluble solute. In other words, it can't actually move through the cell membrane by diffusion, by passivity. It has to come through some type of transport, specialized. These specialized transporters are called blood transporters. So again, let's call here. It's specifically called a blood transport. But let's say that we're having this occur in different organs. Blood transporters are blood is glucose transporter. They're the ones that are bringing this glucose into the cell, but not just one blood in the body. But at the same time, they bring glucose from actually what? Inside the cell, outside the cell. But there's many different types of blood receptors. One way that I like to just easily, quickly remember all of them is a helpful mnemonic in my opinion. It's helpful you guys in this like here. It goes like this. B, B, B. Okay? Kids.
Bidirectional protein transport. Glut.
Lips. R, this doesn't matter, okay? Ink. Mother. Off.
Let me just use something else to play this video. It's a bit distorted.
Which is 2,3 carbon molecules. But the question is, how can we get this glucose, this 6-carbon molecule, into the, let's say that we're having this occur in different organs. Blood transporters are, this glucose transporter, they're the ones that are bringing this glucose into the cell, but not just one direction, but bi-direction. But at the same time, they can bring glucose from actually, what? Inside the cell to outside of the cell. But there's many different types of blood receptors. One way that I like to just easily, quickly remember all of them, it's a helpful mnemonic in my opinion. So you guys know this help here. It goes like this. So like this. B, 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 okay? Kids, lips, R, but this doesn't matter, okay? Ink, mother, pop. Okay, so it's just a little quick way that helps me to basically categorize my blood receptors. And it's going to go in a specific order. So this is actually going to be for blood 1.
Blood brain.
This is going to be for
Blood brain.
blood 2. Ink is going to be for blood 3. And mother and father is going to be
So there are 4
for
types
blood
of
4.
glucose.
Now, what does each one of these things mean? So BBB, the first B I like to remember is blood. But specifically, what type of blood? I mean the red blood cells. So on the red blood cells, plasma membrane, they have a blood receptor. I also like to remember B for baby. And we don't say baby, we say the fetus.
Fetus.
So the fetus is actually going to have blood receptors. Okay? And then the last one is the blood-brain barrier. So blood, brain, barrier. That's the actual sector. connection between the actual vessels and the feed monitor and the middle tissue, the astrocytes around them. We'll talk about that more in the neurophysiology. But again, these are the three easy ones to remember for blood one, BBB. Blood, specifically red blood cells, baby, I like the condition, technical to fetus. And the third one is BBB, blood-brain barrier. Now, what about the second one? So I like to highlight here, AI, I like to highlight the LI, I like to highlight the P and S, right? So what does that do? Well, the KI
Kidneys, liver,
is going to be the
pancreas.
kidney. So the kidney has GLUT2 receptors. The LI is for the liver, and the PS is for the pancreas. There is other tissues. These aren't all of them. Obviously, you can also find within the gastrointestinal tract, there is certain types of GLUT2 receptors also. But anyway, GLUT3, pink. So I like to remember the P right there. The P for placenta. The N for neuron. And another K. Kidney. It's not that bad, right? When you think about it. Again, what is it going to be? It's going to be placenta. And then we're going to have neurons. We're going to have these GLUT3 receptors. Don't get that confused with the actual GLUT1, which is for the blood-brain barrier. Okay, it's different. Okay, because it's going to be where the astrocytes are, right between the blood vessels and the astrocytes. So don't get that confused with the actual neuron cell membrane. Okay, and then K is going to be for again. The N for neuron, and another K. It's not that bad, right? When you think about it. So again, what's it going to be? It's going to be placenta. And we're going to have neurons. We're going to have these GLUT3 receptors. Don't get that confused with the actual GLUT1, which is the blood-brain barrier. Okay, it's different. Okay, because it's going to be where the astrocytes are, right between the blood vessels and the astrocytes. So don't get that confused with the actual neuron cell membrane. Okay, then K is going to be for again. And then the last one. Mother and father. I like to remember N for muscle and F for adipose or fat. So yeah, what is this last one here? Mother and father is going to be specifically just used individually. Mother is for muscle. And father is for fat. Or we should be technical and say adipose. Okay? So the whole purpose of this is just helping you guys to realize that there's many different blood transports. So what they're doing is they're bringing glucose from outside the cell to the inside the cell. But they these ones are generally independent. What does that mean? That means this one. Why I want to say this is because GLUT4 is different from a lot of these other ones. These ones are generally insulin independent. In other words, they don't depend upon the concentration of insulin or their amount. This number, GLUT4, is insulin dependent. What does that mean? That means when insulin is present, he can help to be able to increase the number of GLUT4 transports or increase the number.
We'll go back to something else maybe. Photos will be easier.
The astrocytes. Don't get confused with the actual neurons. There's many different blood transports. Is insulin dependent? What does that mean? That means when insulin is present, he can help to be able to increase the number of GLUT4 transporters or increase the efficiency of the GLUT4 transporters. Whereas GLUT1, GLUT2, GLUT3, they don't really depend upon the presence of insulin. They can function and bring glucose into the cell, out of the cell, and they can regulate based upon the presence of glucose, not by the presence of insulin. Again, that's a quick thing to remember. Okay, so now that we have all of our GLUT transporters and we know exactly how this glucose is actually getting into the cell, now we can move on. Okay, so we bring glucose into the cell through one of these GLUT transporters, depending upon the order. Once we bring it in, we have to chain it. You know what I mean?
And now it's not responding. That's it. Let down. So something to do with the USB itself. Let me just try copying it into the desktop, hopefully. Okay, hold on. Let me just eject the USB itself. Okay, hopefully it's going to play smooth this time.
The Li is for and they can regulate it based upon the presence of glucose, not by the presence of insulin. Again, that's a quick thing. Remember. He can help to be able to increase the number of GLUT4 transporters or increase the efficiency of the GLUT4 transporters. Whereas GLUT1, GLUT2, GLUT3, they don't really depend upon the presence of insulin. They can function and bring in glucose into the cell, out of the cell, and they can regulate it based upon the presence of glucose, not by the presence of insulin. But again, that's a quick thing to remember. Okay, so now that we have all of our GLUT transporters and we know exactly how this glucose is actually getting into the cell, now we can move on. Okay, so we bring glucose into the cell through one of these GLUT transporters, depending upon the order. Once we bring it in, we have to chain it. You know what I mean? So, because remember what I told you, this is actually bidirectional. So at the same time, glucose could move out. To prevent this actual glucose from moving back out, we have to put something on it to prevent it from moving. So what we do is we put a special molecule.
Phosphate.
So let's say that this is carbon number 1, 2, 3, 4, 5, 6. Okay? And on the 6th carbon, I have a special thing coming off. Look at that. That right there is a phosphate. So what did I just put here? I put on here a CO4 3 negative group. Right, that's our phosphate. We put phosphate on the 6th carbon of glucose. So what is it going to be called? This was glucose. There's a phosphate on the 6th carbon of glucose. This must be glucose 6. I'm just going to put P, but you guys were getting one. It's for phosphate. Okay, if I abbreviate some of these, I'll try to explain them. It's just easier to write it down in abbreviations. Okay, now we got this glucose 6 phosphate. The question is, how the heck did that happen? How did I get this no phosphate to a phosphate? There had to be some enzyme involved. Yes, it was. What is the new enzyme involved? This enzyme, it depends upon the tissue that we'll talk about. There's 2 enzymes. One is going to be called 𝐒𝐭𝐮𝐝𝐲. Okay? What is specifically called 𝐒𝐭𝐮𝐝𝐲? The other one is going to be called glucokinase. Now we'll keep this simple. The other one is going to be glucokinase. And what is the difference? Okay. Hexokinase is actually going to be present within the muscles or other different tissue cells. So many different tissue cells, but a lot of it is concentrated in the muscles. Technically, they call this hexokinase type 2. Glucokinase is primarily in the liver. Let's actually put that there. So hexokinase can be many different tissues, like the muscle. It can be a lot of different tissues in different tissue body. But the liver is the only one that has glucokinase, but they also call it hexokinase 4. Okay, that was the first step. And this is what it's doing. It's involved in this step right here. These 2 enzymes, depending upon the tissue, are involved in the conversion of what? Glucose into glucose 6 phosphate. Now the question is, where did that phosphate come from? I'm glad you guys asked. It's coming from taking ATP and converting it into ADP. So what happened? I had 3 phosphates, right? Because that's adenosine triphosphate. Then I went to adenosine diphosphate. That means I lost the phosphate. Where'd it go? Under the 6 carbon glucose. Who is facilitating that? Hexokinase and glucokinase. Next step, we're going to go from this molecule now, right? We're going to go from glucose 6 phosphate to this guy right here. But now look at this. I'm still going to have the phosphate here, okay? That phosphate is still present. The only thing that's different is all I did is I switched a couple different molecules around a little bit. So I switched glucose and then switched its carbonyl form into different kinds. So all I did was I underwent isomerization. So in other words, glucose 6 phosphate and this other one right here, which is called fructose 6 phosphate, have the same number of carbons, same number of hydrogens, same number of oxygens. Primarily, glucose is usually an aldehyde, and fructose is usually the form of a ketone. Okay? So these guys are just interconverting between like an aldehyde and a ketone, but they're just isomerizing. So this step right here, step number 2, this is step number 1. Step number 2 is going to be catalyzed by a phospho, hexose,
isomerase.
Okay, so hexose isomerase. You know, hexose just means 6 carbons. It's a 6 carbon sugar right here. All of this enzyme is doing is converting glucose 6-phosphate into this other guy. What is this guy called? This guy here is called fructose 6-phosphate. So again, what is he called? Fructose 6, and I'm just going to put the P, guys, for phosphate. Okay. That was the second step. Now we're going to go into the third step. We'll talk about this third step in great detail in the other video when we talk about regulation. But in this third step, it's a very important step. Just like this first step is a very, very important step. And you can tell the difference. Why can you tell the difference? This is a black arrow, this is a pink arrow. The pink arrows are reversible steps. So what does that mean? That means not only can I go from glucose 6-phosphate to fructose 6-phosphate, but I could go from fructose 6-phosphate to glucose 6-phosphate. But if I wanted to go from fructose 6-phosphate to this next molecule, that's possible. But I cannot go through that same pathway backwards. This step is not reversible by this enzyme I'm going to mention. You have to move through another enzyme. This is a irreversible step, very regular. Okay, so the enzyme involved in this step is going to be that of phospho, fructokinase 1. And like I said, I'm just going to abbreviate that PFK, which stands for, again, phospho fructokinase type 1. It's involved in this step here. Okay, exactly how is it involved? What it's doing is, if you notice, I'm gonna show you something here. This was the 6 carbon, this was the 1 carbon, right? I need you guys to do the math. 2, 3, 4, 5. On the 6 carbon, we still have that phosphate. Nothing has changed there. And now it's going to be on the 1 carbon. So now we have a phosphate on the 6 carbon, and we have a phosphate on the 1 carbon. What must this molecule be called? There's a phosphate on the 6, there's a phosphate on the 1st. It should be called fructose 1,6 bisphosphate. Why do you call it bisphosphate? Because you've probably heard the term biphosphate and bisphosphate. Bisphosphate means that there's carbon spaces between it. Biphosphate means that they're right next to one another. Okay? So bisphospho means that the actual phosphate groups are actually separated. They're a couple carbons away. Or biphospho means that they're right next to one another. Okay? But in this case, it's bis. Okay, so what is this molecule called? Fructose 1,6. I'm going to put BP4, bisphosphate. Okay, what is the enzyme involved in that step? The PFK1, right? What happened? I had 1 phosphate originally on the 6 carbon, then I added another. Oh, I must have done the same thing that I did in this step here. Yep. So ATP is involved in this step here. And he uses a phosphate that's converted into ADP. So ATP is converted into ADP. Okay, cool. So I actually used up an ATP in that step. Now, look what happens here. I take this fructose 1,6 bisphosphate, which is 6 carbons, 1, 2, 3, 4, 5, 6, and I split it in half to 2 3-carbon fragments. Okay? But the phosphate on this carbon right here should be on this carbon here. So now let's put a phosphate over here. Okay? And then that phosphate that's going to be over here, it should be right there. Alright, cool. And again, what is this guy right here? This is a phosphate. And again, what is this guy right here called? This is a phosphate. Okay. Now that we've done that, what are these guys called? Now these ones are a little bit harder to name, okay? Unfortunately. I'm going to give you guys a mnemonic at the end that will help you a little bit with that. But this guy right here is called dihydroxyacetone phosphate. Okay? So it's a 3-carbon molecule with a ketone in the middle and a phosphate on that carbon. Okay, we can technically call it the 1 carbon, but it wouldn't matter because on either side, no matter how you name it, nomenclature-wise, it doesn't matter. The dihydroxyacetone phosphate. Okay, there's that guy. Now, the thing is, dihydroxyacetone phosphate isn't really utilized in this actual glycolysis pathway. He has to be converted into another molecule. What's this molecule here called? This one right here is different from this guy. This one is actually having an aldehyde on the end, so we call him glyceraldehyde. Glyceraldehyde 3-phosphate. Okay? Sometimes you might even see, like I'm actually going to denote this one as DHAP, dihydroxyacetone phosphate. And this one you're probably going to see in other videos as GA3P. Glyceraldehyde 3-phosphate. Okay? Now, here's the thing. What enzyme was involved in splitting this puppy? Okay? They call this enzyme involved in this step here. Right? Or we can even say, if you want to, involved in this step here. The same enzyme, it's just cleaving. This enzyme is called aldolase. It'll actually go like this instead. Just kind of encase around this. Circle this. He's involved in both of these steps. So he's actually doing what? He's cleaving fructose 1,6-phosphate to what? DHAP and GA3P. Now, like I told you, dihydroxyacetone phosphate doesn't actually convert into this next glycolytic thing. He has to be converted into glyceraldehyde 3-phosphate in order for him to be converted into this next thing. So we have to have an enzyme that allows for this interconversion or isomerization. That enzyme that's involved in this step is called a triose, because it's a 3-carbon molecule. Phosphate, I'm going to put P, isomerase. It's called a triose phosphate isomerase enzyme. And it's involved in the interconversion between GA3P and DHAP, or DHAP to GAP. Okay? Primarily, with a little bit more of it going towards the GA3P, depending on the body's name.
Glyceraldehyde 3-phosphate.
The enzyme is called triose.
P, or isomerization. That enzyme that is involved in this step is called a triose, because it's a 3-carbon molecule. Phosphate, I'm going to put P, isomerase.
Triphosphate isomerase.
So it's called a triose phosphate isomerase enzyme. And it's involved in the interconversion between GA3P and DHAP, or DHAP to GAP. Okay?
4.5.
Primarily with a little bit more of it going towards the GA3P.
Let's say it's a 4.5 step, but half
Let's
step,
talk about that for a minute. Now,
not a full step.
glyceraldehyde 3-phosphate, what happens to him? He is then going to get converted to this next guy here. Okay, well this guy here has a phosphate on what carbon? So let's number this carbon. 1, 2, 3. What happens to him? He is then going to get converted to this next guy. A little bit more of it going towards the GA3P depending on the body. Alright, we'll talk about that in other videos. Now, glyceraldehyde 3-phosphate, what happens to him? He is then going to get converted to this next guy here. Okay, well this guy, well then it should still have a phosphate here, I would suppose, right? Let's see. Okay, it has a phosphate here. Oh, GA3P, DHAP, or GA. Or DHAP to GAP. Okay? Primarily with a little bit more of it going towards the converted to this next guy here. Okay, well this guy here has a phosphate on what carbon? So let's number this carbon. 1, 2, 3. Okay, that means that the phosphate is on the 3rd carbon. That's why we named it that. Well then it should still have a phosphate here, I would suppose, right? Let's see. Okay, and it has a phosphate here. Oh, would you look at that? There's another phosphate. Huh. I added another phosphate. Okay, that's cool. Now, what? Positives in this step. Okay, so I'm going to show it coming. This enzyme is working right here in this step. Now I'm going to kind of abbreviate him also. He is called glyceraldehyde 3-phosphate, just like that, but dehydrogenase. Okay. Well, this guy here has a phosphate on what carbon? So let's number this carbon. 1.
This is a big of a letdown now, since I can't properly play the video. Let me check. This way. Hopefully
And this
it
step
does.
is called a triose,
Okay.
because it's a 3-carbon molecule. Phosphate, I'm going to put P. Phosphate on what carbon? So let's number this carbon. 1, 2, 3. Okay, well, that means that the phosphate is on the 3rd carbon. That's why we named it that. Well, then it should still have a phosphate here, I would suppose, right? Let's see. Okay, and it has a phosphate here. Oh, would you look at that? There's another phosphate. Huh. I added another phosphate. Okay, that's cool. Now, what had to happen then? And what would we call this molecule here? We would actually call this molecule specifically 1,3. Now, because the phosphates are actually having a space between them, this is bis phospho, and then it's actually, this is the 3-carbon glycerate. Okay, so it's called 1,3 bis phosphoglycerate. That's this molecule. Now there's a special enzyme involved in this step. It's a pretty cool enzyme. This enzyme is working right here in this step. And I'm going to kind of abbreviate him also. He is called glyceraldehyde 3-phosphate, just like that, but dehydrogenase. Okay, this is an important step. Okay, a very important step here. What's going to happen is this enzyme, this glyceraldehyde 3-phosphate dehydrogenase, he's going to do two things. He's going to add two things into this reaction. One thing I'm going to do is I'm going to take NAD positives in this step. Okay, so I'm going to show it coming off of this reaction here. So look, I'm going to have NAD positive. He's going to react with that glyceraldehyde 3-phosphate in the presence of this enzyme. And what he's going to do is he's going to rip off hydrides off of the GH3. And when he does, he gets converted into NADH. What is hydrides? Hydride is just a fancy way of saying, here's my hydrogen, and then this hydrogen, it's going to have a proton, within the center of it, and then what's going to be around it generally has just one electron. But in this case, a hydride has 2 electrons. What do we say a hydride is technically? We say a hydride is really a proton plus 2 electrons. Okay? That's what's going to be our hydride. And that's what this NAD molecule, this NAD plus, is doing. It's picking up hydrides and getting converted into NADH. Here's the tricky thing though. Look at this. I want you guys to really, really see this. This is fructose 1,6-bisphosphate. It gets converted into DHAP and GA3P. I told you most of this guy is getting shifted over here. So how many of them do I really actually have? I have 2. Okay? I have 2 of him. Because I told you, most of this is being shunted over here. And then he's running through this reaction twice. So if that's happening twice, then how many NADs am I actually producing? I'm actually producing 2. I'm having 2 NADHs. Okay, cool. That doesn't account for this phosphate. This enzyme's so tricky. He loves to just add in an inorganic phosphate. So you see here I'm going to have an inorganic phosphate. I'm just going to throw that into the reaction. So GA3P, dehydrogenase, is going to just throw a phosphate into the reaction and generate NADHs. Okay, cool. So we're good with that. Now we're going to the next step. Now look what happens here. You're going to notice something different. Start off there. What does that mean? That means there's a loss of phosphates. But again, what would we call this one? Okay, well, it's going to have a similar name, but just get rid of a 1. The 3, but no biz, it's just like 3 phosphoglycerate. What are we going to call this? 3 phosphoglycerate. Okay, that's the name of this substrate. So now what is this one here called? 3 phosphoglycerate. Now, in this step, there's a special enzyme involved here. This enzyme is really cool, and what he's doing is he's actually going to help to form, let's do this nice in color here. I'm going to take ADP and convert it in this step to ATP. Okay? But because this reaction is happening twice, how many am I actually? Producing, guys, 2. Okay, so I'm actually producing 2 ATP in this step right here. Okay, this enzyme is pretty cool. This enzyme right here that's working this step is called phosphoglycerate kinase. Okay, so phosphoglycerate kinase is really, really special because what he's doing is, what's the definition of a kinase? He talks about it.
Kinase is just adding phosphate.
By definition, something that phosphorylates a substrate. Okay, well, what does it phosphorylate? It's phosphorylating ADP. Where's it getting that phosphate from?
Phosphoglycerate kinase.
the 1,3-VPG. It's ripping off of the one carbon and giving it to ADP to make ATP. So that's what he is involved in. He is involved in this step here. Okay. Good. We got our 2 ATP. All right, that's cool. Okay, so now. There's 3-phosphoglycerol. Nothing crazy is going to happen in this next reaction. It's nothing, it's not a really special, reaction. You can see. Look what happened to the phosphate. All I did was I switched it. I mutated it and I switched it from the 3rd carbon to the 2nd carbon. Because again, we denote this 1, 2, 3. Same thing here. 1, 2, 3. So I switched it to the 2nd carbon. So what would you guys suppose this is going to be this guy's name? 2-phosphoglycerol. Nothing crazy in this one. 2-phosphoglycerol. Okay, cool. Now the enzyme involved in this one's not really that important, but we'll mention it anyway, just that you guys can have that. The enzyme involved in this step, converting the 3-phosphoglycerate into the 2-phosphoglycerate, is just called phosphoglycerate mutase. So phosphoglycerate mutase. Okay, that's that enzyme. He's stimulating this stuff. Okay, cool. Now, the 2-phosphoglycerate is going to go to a kind of an interesting step. Okay, so what he's going to do is, this phosphate is on the carbon, right? So like, let's say that here I have a 3-carbon structure, just for a second. I have a 3-carbon structure. And then what I have here is I have the phosphate coming off. What I'm going to do is, I'm going to switch some structures around, and I'm going to convert this into what's called an enol. And an enol is just when you have, according to organic chemistry, it's a double bond between these carbons with an alcohol coming off like that. That's an enol. But what I'm going to do is I'm going to put the phosphate on that now. Okay, so I'm going to have it kind of like switched off a little bit. So because I have this different kind of structure, what I'm now going to have is I'm going to have this phospho enol, and it's a 3-carbon structure like this last one, pyruvate. What is this last molecule here called? We call this molecule phosphoenol pyruvate. This one here is called phosphoenol pyruvate. But I don't like that. I like PEP. Okay, it's easier to remember. Okay, but it's, it's up to you. Alright, so PEP. And what am I doing? All I'm doing is I'm shifting it into a different kind of structure. So it's just making it a little bit more modified now. Okay, and then again, this is going to be my phosphate. So what do I call this? Phosphoenol pyruvate. The enzyme that's doing that is trying to convert it into an enol. So it's called an enol. So what is the enzyme involved in this reaction? The enzyme involved in this reaction is called a enase. Okay? Nothing crazy. Really the more important steps in this process is the black lines one that I mentioned, as well as this reaction. Okay, last one. I'm taking the phosphoenol pyruvate and look what happens. Meaning no oxygen. Very, very little oxygen. And over here, this is when you aerobic. Meaning that you have oxygen.
Yeah, in anaerobic it converts into lactate, and in aerobic it converts into, goes into the TCA cycle, converted into carbon, hydrogen, carbon dioxide, water, and ATP is formed. Okay, I guess that's it for it was just a very short stream. I'll come back to these notes hopefully one by one. I'll try to, later. Okay, we're back. So okay, so yeah, I'll just try to put these notes tomorrow in the Obsidian, and that's it for today. Inshallah, the upcoming streams will be longer. Hopefully, we're going to have some really good study sessions. Inshallah. That's it. Assalamu alaikum wa rahmatullahi wa barakatuh.